Hello and welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. Guys, today we're going to have a short lecture on satellite cells and how that applies to our muscles and muscle memory. So if that's what you're here to find out about, then in a short, sweet, science-based video, let's jump right into it. All right, let's jump into this. So if you look at this diagram that I've got up here, obviously this is probably not going to be recognizable to you unless you know what a myocyte looks like, a muscle cell looks like. And that's essentially what we're looking at. We're looking at just a particular section of a muscle cell. So if you extend your arm out and you look at your arm, whenever you're moving your arm, those myocytes are moving in and out. So they're kind of contracting and extending. And if we zoom into your arm, you're going to see many millions of these types of cells that extend from your elbow all the way up to your shoulder. And then you have insertions, but that's beside the point. So on this myocyte, on this muscle cell, what you find is that what's unique, and pretty unique, not many other cells have this, is that they have multiple nuclei. So the nuclei, or the nucleus, is where all your DNA is kept, or the majority of your DNA is kept. And that is kind of the initial site of where you're going to have protein synthesis occur. Not the actual synthesis itself, but you're going to get these genes that are going to be expressed to then create mRNA, and then from mRNA templates, then you can create proteins, which are then integrated into this muscle cell. So what's really interesting about this is that with all of these nuclei, there's something called a myonuclear domain, in which a single nucleus on a muscle cell, although there are many different nuclei, a single nucleus will be essentially responsible for a particular space around it. So it's responsible for creating enough proteins to maintain that area. Now, if you end up stimulating that area, if you end up incurring some sort of damage, then in that situation, this is poorly signified by me by these red squigglies here, which are essentially just damage that's occurring on the sarcolemma, which is the membrane of the cell. Then you have a drive to integrate more myonuclei, to create more of these nuclei, because eventually this fiber is going to extend outwards as it starts to hypertrophy, it starts to push outwards. And that's why you go from a state of being really skinny and having almost no muscle to suddenly becoming really jacked. Then suddenly you have this increase in size, and that's because you have muscle hypertrophy in the myocytes themselves. So for that to be able to be possible, you have to have, with this damage, you have to have an integration of new myonuclei. So not only are the present myonuclei suddenly upregulated and they have to work really hard to keep expressing particular genes to get those proteins created, but you also have to have this integration of new ones. So where are these new ones coming from? Well, if you heard the intro to this video, you know that that's going to come from these green things. These green things are what are called satellite cells, and they kind of rest on top of your myocytes, your muscle cells. And when you have this damage that occurs on the sarcolemma, you have these satellite cells that can then migrate their way into the muscle cell itself. And then what they do is, because they're primarily nucleus, they will integrate their nucleus into the muscle cell, which then leads to more myonuclei. And then with that increase in these myonuclei, then you have a diminished myonuclear domain. So then you can then have that one be responsible for what would have been responsible by some of these other myonuclei. So you have more myonuclei leads to a lower threshold, or a lower expression, a smaller area that it's responsible for, and then it's going to continue as the muscle continues to grow. That is going to enlarge, enlarge, enlarge until you get to a point where you need more myonuclei, and they keep integrating. Now, how that plays into with the actual muscle memory is where you have these myonuclei don't go away. So let's say you were to stop exercising, stop stimulating that musculature for, let's say, 6 months, 10 months, whatever it ends up being, and you see this myocyte then starts to shrink back to where it was. These myonuclei, they're fully integrated. They can't move back out. So they're stuck in place, which is a high benefit because the DNA that's found inside is going to remain, meaning that then if you were to re-stimulate this area, you don't have to go through this process of reintegrating these satellite cells. All you have to do is, or all your cells have to do, is just re-stimulate these already integrated myonuclei, and then you get more protein synthetic response as a result. So there you go. That's one mechanism and a really well characterized, well understood, well, relatively well understood characterization of essentially muscle memory. And I'll be presenting on more muscle memory mechanisms in the future. All right, with that said, hope I have the pleasure of speaking with you next time. Have a good one. Bye.